Alright, ladies and gentlemen, are you there? I want your attention, please. Alright, the presentation is about to begin, alright? And I want you, all of you, to be in your camera and your computer and whatsoever, to get ready and well prepared for our next speakers, alright? Alright, ladies and gentlemen, and now we all gear up. Why don't we make this afternoon not just wonderful, but also a day full of learning and sharing? Alright, so please be reminded, if you have any question to ask during any session, type your questions in the comment box below the session video and click post comment button. Alright, and we will try to get question to be answered by the community. Alright, and I will pass to my partner Iza for the next agenda. Alright, Iza.
Hello and good evening. Thank you to each and every one of you for being and stay here with us today. I hope you enjoy your lunch. Without further delay, let's hear out our next young and beautiful ladies, Izzah Jalal from University Kebangsaan Malaysia, on her opinion in Tasik Chini issue. Please welcome.
Hello and good evening. My name is Nurul Izzah binti Jalal and I'm a student from National University of Malaysia and currently pursuing a degree in biochemistry. So the topic I chose today is self-awareness regarding the Biosphere Reserve of Tasik Chini and its importance. So to start off, as we all know, the Earth consists of 4 spheres, which is the geosphere, the hydrosphere, the atmosphere, and also the biosphere. So we're going to talk about the biosphere, and to put it in a simple term, the biosphere is actually made out of different biomes, in which biomes are plants, animals, and other living things that share similarities in the environment they live in. So for example, forests, rainforests, deserts, and also wetlands are types of biomes that make up our biosphere. So Biosphere Reserve, on the other hand, is protected area and was actually initiated back in 1975 by UNESCO under a program named Man and Biosphere, MAB. So Biosphere Reserve consists of 3 zones, which is the core zone, the buffet zone, and also the manipulation zone. So these zones are categorized in which what kind of activities are allowed. So for example, the core zone, no human activities are allowed, while the buffet zone, minimal human activities such as research and also education are allowed. However, for the manipulation zone, human activities such as cropping and recreation are allowed as long as it does not disturb the environment. So Tasik Chini is actually one of Malaysia's first biosphere reserve that was granted by UNESCO back in 2009. However, Tasik Chini may lose its status because back in 2019, unregulated land mining and also clearing has been done, causing the lake to overrun with mud, making the clear waters turn cloudy and also fewer fish, and also there are making few tourists visit that area. So if this continues, the ecosystem could potentially collapse by the year 2030. So why do we need to have and also protect our biosphere reserve is because it conserves the genetic resources, species, ecosystems, and landscape without uprooting inhabitants, as well as biosphere reserves actually helps to rebuild any damage caused to ecosystem and also a habitat. And most of all, in a biosphere reserve, endangered species of animals and plants are protected. So to put it in a nutshell, I urge that we all get to know our biosphere and take part in protecting it. Thank you.
Thank you very much, Izzah Jalal, for your sharing on self-awareness regarding the Biosphere Reserve of Tasik Chini and its importance. Alright, ladies and gentlemen, please stay with me for our next session, Without any further delay, I would like to invite our next keynote speaker, Yang Berbahagia Datuk Seri Dr. Salihuddin bin Ishaq, Secretary of State of Pahang. He will give a speech on the role of the state government in maintaining the biosphere reserve site. Please welcome. Tuan-tuan dan puan-puan sekalian, majlis seterusnya menjemput dan mempersilakan Yang Berbahagia Datuk Seri Dr. Salihuddin bin Ishaq, Setiausaha Kerajaan Negeri Pahang, untuk memberikan ucapan berkaitan dengan peranan kerajaan negeri dalam mengekalkan tapak rizab biosfera. Majlis mempersilakan.
Assalamualaikum warahmatullahi wabarakatuh dan salam sejahtera. Alhamdulillah, bersyukur saya ke hadrat ilahi kerana dapat kita semua berkumpul secara dalam talian pada hari ini dalam persidangan antarabangsa sains dan rizab biosfera yang julung-julung kali diadakan. Meskipun dengan pandemik COVID-19 yang melanda dunia pada ketika ini, tetapi dengan kepantasan teknologi, acara seperti ini dapat juga diadakan. Saya mengucapkan syabas dan tahniah kepada penganjur serta seluruh ahli jawatan kuasa persidangan antarabangsa sains dan rizab biosfera serta staf Pusat Penyelidikan Tasik Chini yang telah berusaha dalam merencanakan persidangan ini. Saya amat percaya bahawa persidangan ini akan menjadi platform diskusi secara akademik antara penyelidik tempatan dan antarabangsa, juga agensi-agensi kerjaan dan pemain industri dalam bidang sains dan teknologi alam sekitar. Seperti termaktub di dalam rancangan struktur negeri Pahang, Pahang adalah nafas negara kerana fungsi negeri ini yang menjadi penyumbang utama untuk mengekalkan alam semula jadi yang menjadi pembekal kepada oksigen negara melalui pengekalan hutan hujan tropika yang dikatakan antara tertua di dunia. Juga kerana kesediaan negeri Pahang untuk mengekalkan kawasan yang dilitupi hutan. Kesediaan mengekalkan hutan memerlukan perkongsian dalam berbagai aspek dan kepakaran, pengurusan teknologi, pemeliharaan, penguasaan dan tanggungjawab keuangan. Sebagai destinasi pelancongan terunggul, negeri Pahang cukup sinonim dengan kekayaan alam semula jadi seperti hutan hujan tropika tertua di dunia, pantai dan pulau, taman-taman laut, wetlands bertaraf dunia dan tasik-tasik air tawar. Justru pelaksanaan rancangan struktur negeri Pahang melalui cadangan dan inisiatif pemeliharaan sumber semula jadi menaik taraf kemudahan dan kemudahansampaian serta penambahbaikan produk dan perkhidmatan pelancongan serta pembinaan struktur dan menambah baik akan meningkatkan lagi tarikan pelancong datang ke negeri ini. Visi rancangan struktur negeri Pahang 2050 adalah untuk mencapai Pahang Cemerlang 2050. Visi ini berlandaskan 4 teras utama. Pertama, spasial seimbang, maju, sejahtera, serta perlindungan dan pemeliharaan. Spasial seimbang menekankan perancangan dan guna ruang yang efisien dan bersepadu. Teras kedua adalah maju. Ini menjurus kepada pertumbuhan ekonomi stabil dan berdaya tahan. Teras ketiga pula ialah sejahtera yang mementingkan komuniti inklusif dan persekitaran berdaya serta berkualiti. Teras yang terakhir ialah perlindungan dan pemuliharaan yang melibatkan khazanah alam semula jadi, habitat dan kawasan sensitif alam sekitar. Semua sedia maklum bahawa Tasik Chini adalah khazanah negara kerana ia adalah tasik air tawar semula jadi yang kedua terbesar di Malaysia. Bukan sahaja kaya dengan kepelbagaian biodiversitinya, Tasik Chini juga menyimpan nilai estetika yang tinggi yang merupakan tempat bertapaknya aktiviti-aktiviti ekonomi dahulu, seperti menangkap ikan secara tradisional dan mencari hasil hutan. Selain menjadi tempat tinggal bagi penduduk zaman dahulu kala, kerana bekalan airnya yang terjamin, Tasik Chini juga bertindak sebagai kolam takungan bagi menampung lebihan air pada musim monsun. Pada Mei 2009, Tasik Chini telah mendapat penganugerahan sebagai tapak Rizab Biosfera melalui program Man and Biosphere yang telah dilancarkan oleh Pertubuhan Pendidikan, Sains dan Kebudayaan Pertubuhan Bangsa-Bangsa Bersatu atau UNESCO. Justru, Kerajaan Negeri Pahang amat komited di dalam menjalankan usaha dalam mengekalkan kelangsungan Tasik Chini. Salah satu inisiatif yang telah dirangka oleh Kerajaan Pahang ialah mewartakan kawasan seluas 4,498 hektar di sekitar Tasik Chini sebagai taman negeri. Inisiatif ini menjadi titik pemangkin yang menunjukkan kesediaan Kerajaan Negeri Pahang dalam mengekalkan khazanah negara yang tidak ternilai ini dan dalam masa yang sama memanfaatkan potensi ekonomi di kawasan tersebut. Usaha pemulihan dan pemeliharaan persekitaran Tasik Chini juga sedang giat dilaksanakan melalui beberapa agensi kerajaan. Antaranya, Jabatan Alam Sekitar Negeri Pahang menjalankan persempelan air secara bulanan bagi memantau kualiti air tasik. Jabatan Pengairan dan Saliran Negeri Pahang pula menjalankan aktiviti pembersihan di sungai-sungai yang berdekatan dengan tasik. Di samping itu, Jabatan Perhutanan Negeri Pahang telah memperuntukkan sebanyak RM50,000 pada tahun ini bagi menghutankan semula kawasan sekitar Tasik Chini. Diharapkan dengan usaha ini, kita akan mengembalikan Tasik Chini sebagai destinasi eko pelancongan yang tersohor di negara yang bertaraf dunia. Daripada hasil aktiviti ini semua akan dapat digunakan untuk melindungi kepentingan biodiversiti di samping kesejahteraan masyarakat Tasik Chini dan di kawasan sekitarnya. Mengenai perlombongan, Kerajaan Pahang mempunyai pendirian yang tegas bahawa tiada lagi kelulusan perlombongan yang baru di kawasan Tasik Chini, manakala lesen-lesen yang sedia ada sentiasa dikawal dengan ketat dan juga dipantau pada setiap masa. Akhir sekali, Kerajaan Negeri Pahang amat menyokong usaha-usaha ahli-ahli akademik termasuk Universiti Kebangsaan Malaysia dan Pusat Penyelidikan Tasik Chini yang sentiasa berusaha dan juga menganjurkan persidangan antarabangsa ini bagi menjadi wadah perkongsian hasil kajian di dalam usaha mengekalkan tapak Rizab Biosfera dan juga menyebarluaskan kepentingan pemulihan dan pemeliharaan Tasik Chini untuk melonjakkan kembali kegemilangan Tasik sebagai pusat pelancongan domestik yang bertaraf antarabangsa. Buat akhirnya, selamat bersidang. Wassalamualaikum warahmatullahi wabarakatuh.
A huge thanks to our honored keynote speaker, YB Datuk Seri Salahuddin bin Ishaq, Secretary of State of Pahang. We hope the talk would show us a clear direction of Pahang State Forestry Department in order to conserve Tasik Chini as Biosphere Reserve. Let us straight away with our next two schoolboy students with their thought on our work, our responsibility. Please welcome Adik Muhammad Nurul Syawan bin Ismail and Muhammad Lutfi bin Abdullah from Mahat Tafiz Al-Quran, Tok Montok.
Assalamualaikum, my name is Muhammad Lutfi. I'm 13 years old.
Assalamualaikum, my name is Muhammad Syawal. I am 15 years old.
Dear nature, you're probably wondering when we've all gone. Where are all those people who were wandering in your woods? Where are all those airplanes that were flying in your skies? Isn't all this silence strange? Dear nature, we miss you. Probably more than you miss us. Take this time to breathe, to bloom. To conquer your space. And when you're back, we promise to treat you better than we ever did before. Because now that we're so distant from you, we truly understand how much you really mean to us. I love you, nature.
Terima kasih diucapkan kepada Syawal dan Luffy di atas perbentangan itu tadi. We love you too, nature. It is blessed to have the young generation that truly understand how much the world really meant to us. Melentur buluh biarlah dari rebung. Alright, ladies and gentlemen, without waiting any much longer, I would like to welcome our next presenter, Miss Nur Afrina Batrisha from Universiti Kebangsaan Malaysia on her research Chlorella species and Moina macrocopa as feeds for sustainable aquaculture in Tasik Chini. Please welcome.
Hello and good afternoon or evening to speakers, fellow presenters and spectators. Today, I, Afrina Badesha, a student under Dr. Muhammad Shazul Faziz Arijaya, will be presenting today about my research titled Chlorella species and Moina macrocopa as feeds for sustainable aquaculture in Tasik Chini. Valuable fish stocks as well as other marine life are severely threatened by overfishing. According to World Wide Fund for Nature, people are taking far more fish out of the ocean than can be replaced by those remaining. This is why aquaculture is becoming economically more important as a way of increasing local fish production for food security and to improve livelihoods. Aquaculture can be defined as the farming of aquatic organisms including fish, mollusks, crustaceans and aquatic plants. Nutrition and feeding plays an essential role in sustained development of aquaculture. However, aquaculture production is produced by intensive farming system using high-cost nutrient inputs in the form of nutritionally formulated diet. 30 to 50% of commercial feeds are produced by unsustainable fishing. In this research, we aim to introduce, especially to local fish farmers, a sustainable alternative form of feed resources instead of formulated fish diet. Feeding habit of fishes in natural water bodies is different among the species, but all the fishes require protein-rich live food for their better growth, efficient breeding, and survival. Live food can be defined as organisms that are able to swim in water column, constantly available to fish and shellfish larvae, and are likely to stimulate larval feeding response. Nowadays, fresh and live feeds have been favored due to their nutritional values compared to formulated food pelleted. These live foods can be purchased from the market, which is costly and may not be available as and where required. Culturing them economically and sustainably have always been challenged. For example, several parameters need to be taken into consideration when hatching out artemiasis, which is a more well-known live food among fish farmers. Artemia needs a constant temperature between 25 to 28 degrees Celsius, salinity of 15 to 35 ppt, oxygen level close to saturation, and a strong illumination of the seas at the water surface. So, not only we aim to introduce live food as main diets in aquaculture production, we also aim to introduce Moina, macrocopa, and macroalgae as a cheaper and easy alternative compared in using Artemia. Moina macrocopa is a small freshwater cladistral crustacean commonly called water fleas. It has a body consisting of a head and a trunk. The brood pouch, where the eggs and embryos develop, is on the dorsal side of the female. Moina is generally quite tolerant of poor water quality. It can be found living in the water with dissolved oxygen between 0 to supersaturation. It is resistant to change in oxygen concentrations and to extreme temperature. The reproductive cycle of Moina has both asexual and asexual phase. Normally, the population consists of all females that are reproducing asexually. Under adverse environmental conditions, males are produced and sexual reproduction occurs resulting in resting eggs. Moina appear in high concentrations in ponds, lakes, ditches, slow-moving streams, and swamps. Often reproduced in large quantities in water bodies strongly polluted with sewage. Moina feed on various groups of bacteria, yeast, phytoplankton, and detritus. The protein content of Moina usually average 50% of the dry weight, and adults normally have a higher fat content than juveniles. This is approximate analysis of Moina macrocopa. From the graph, between the three nutrient content, we can see that Moina macrocopa has a high protein content compared to fat and carbohydrates. Macroalgae can be defined as a large and diverse group of aquatic organisms that lack the complex cell structures found in higher plants. It can be categorized into marine macroalgae and freshwater macroalgae. Apart from the basic nutrients, algae are also abundant in vitamin and minerals. Macroalgae is suitable as a food resources for aquaculture organisms such as mollusks, fish species, and their larvae, crustaceans, and zooplankton. For culturing in lab, three types of medium are used, which are commercial medium KW21, VG11, and Bristol. It is to see if there is any difference in macroalgae growth using the three different medium. The temperature is set at 22 degrees Celsius and the culture is aerated using air pump and exposed under fluorescent light for 24 hours to achieve maximum growth rate. The microalgae culture is inoculated in a small test tube and periodically will be transferred to a much bigger container. This graph shows the concentrations of chlorophyll A between KW21, BG11, and Bristol. In lab phase, we can see that KW21 has the highest increase of chlorophyll A concentrations from day 1 to day 2. KW21 has 132.57 increased percentage of chlorophyll A concentration, while BG11 has 79.85 increased percentage, and Bristol only has 53.32 percentage. This is due to the higher number of constituents in KW21 that is essential to chlorella species growth. In third day to fifth day, which is the linear phase and stationary phase, you can see that all three mediums recorded a not so much difference value in chlorophyll A concentration. This graph shows the value of salinity of microalgae biomass culture from three different mediums. Microalgae biomass culture from medium Bristol and BG11 show a value of salinity between 0.07 to 0.09. While microalgae biomass culture from KW21 recorded a much higher value of salinity. However, all three macroalgae biomass are all under 0.5 ppt, which is the salinity value of fresh water. Moina macrocopa will be cultured using the batch culture method, where the moina will be inoculated into a 10-liter container, and then after 4 to 5 days will be harvested. The harvested moina will then inoculated into a much bigger tank or container to produce a much bigger moina biomass. This process then will be repeated so that the moina population will be not run out. To investigate moina reproduction, growth and populations, females containing embryos were isolated from a reproductive stock. After the juvenile were released, the juvenile will be used for experiment. Each neonate was put in a test tube containing dechlorinated water and was fed by microalgae daily. Moina was observed and counted daily. From this bar graph, we can see that moina reproduce as early as 4th day after being inoculated, and then if observed, if food resource is sufficient, population of moina will increase day by day. To know the bird size and bird count, one neonate was put in test tube containing dechlorinated water. Once the neonate matured and began reproduce, bird size, which is the number of neonates produced by one adult moina, was determined. Neonates will then be separated from the mother. This procedure was repeated until the female died. Moina was fed daily with microalgae. From the bar graph, we can see that the highest number of bird size is 30, while the lowest number is 1. And the highest number of bird count is 10, which means that the moina produced for 10 days before it died. 15 moina was inoculated in 1 liter of Flora species cultured from 3 different medium. 10 milliliter from the water was taken and populations of moina inside was determined daily. We can see that by 4th day that the populations of moina has increased, and by 7th and 8th day populations of moina starting to decrease. This is because of insufficient food resource as most of the microalgae has been consumed. This picture shows the microalgae culture before and after inoculations of moina. You can see that after 5 days, most of the microalgae has been consumed by moina, leaving a transparent liquid. This graph shows the concentrations of chlorophyll A before and after being fed to moina macrocopa. Moina was inoculated at the 5th day after the microalgae is cultured, and at the 4th day after the inoculations, we can see the populations of moina has increased. However, the concentrations of chlorophyll A is still high. The remaining of the moina was left inside the culture to feed the rest of the microalgae, and at 4th day after the first harvest, the concentrations of chlorophyll A has decreased and the total moina harvest is 2.97 grams. This second graph also shows the concentrations of chlorophyll A before and after being fed to moina macrocopa in 10 liter container. The amount of moina inoculated is much higher than in the first graph. After 4 days, the concentrations of chlorophyll A has decreased and the total amount of moina harvested is 1.43 gram. This shows that the higher the amount of moina inoculated into the microalgae culture, the faster that we can harvest the moina as early as 4 days after being inoculated. This picture shows the microalgae culture before and after inoculations of moina. We can see that by 5th day, most of the microalgae culture has been consumed by moina macrocopa. In conclusions, moina is easy to culture and maintain as it is more resistant to any changes that occur to its surrounding. It has a high reproduction rate and can reproduce as early as 4 days, high protein content and suitable to be used as feed resources to aquaculture production, especially in larval stage. Can be used as source of income as moina production as live can be considered as new. Low initial production cost as a small amount of moina can be used to culture bigger moina biomass.
Thank you very much, Ms. Nur Afrina Batrisha, for your presentations. We hope moina production might contribute as an alternative for livestock and reduce our dependence from import. Alright? At this time, I would like to invite our next honorable keynote speaker to give a speech on topic local community issues and expectations. Ladies and gentlemen, let's welcome YB Muhammad Syahrim Haji Madzin, Ahli Dewan Undangan Negeri Adun Cini Pahang, untuk memberikan ucapannya berkaitan dengan isu-isu masyarakat setempat dan harapan. Untuk itu, majlis mempersilakan.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh dan salam sejahtera. Pertamanya saya mengucapkan syukur kehadirat Allah SWT pada hari ini dapat saya menyampaikan satu ucap tama bagi persidangan antarabangsa sains dan rizab biosfera. Saya amat bersyukur pada hari ini kerana kita semua dapat bersama-sama menyertai persidangan antarabangsa secara maya bagi meneruskan perkongsian ilmu meskipun dalam situasi negara kita dilanda pandemik COVID-19. Pertama sekali saya ingin mengucapkan syabas kepada jawatankuasa persidangan ini yang terdiri daripada ahli akademik UKM dan kaki tangan pusat penyelidikan Tasik Chini yang sangat cakna dengan perkara yang melibatkan Tasik Chini. Bukan hanya memfokuskan perihal Tasik Chini, persidangan ini akan menjadi platform perbincangan menggalakkan penyebaran maklumat dan ilmu sebagai penggalak kepada kerjasama pihak-pihak berkepentingan seperti agensi kerajaan, badan berkanun, universiti dan industri yang memastikan kemapanan salah satu lagi band biodiversity global iaitu Tasik Chini. Diharap platform ini dapat menjadi wadah perkongsian pertukaran ilmu dan pendapat berkenaan dengan penyelidikan sains dan teknologi yang menekankan kesinambungan alam sekitar. Dengan penlibatan pihak Suruhanjaya Kebangsaan UNESCO Malaysia dan juga UNESCO Jakarta, secara tidak langsung persidangan ini dapat mempromosikan keistimewaan Tasik Chini di peringkat masyarakat antarabangsa. Meskipun sektor pelancongan mengalami pengurangan ekoran pandemik COVID-19 di seluruh negara, diharap persidangan ini akan menjadi satu loncatan yang menggalakkan kerjasama antara pemain industri dan juga pihak agensi kerajaan. Dalam transformasi ekonomi global yang menjurus kepada pembangunan ekonomi masyarakat setempat, komuniti minoriti seperti orang asli tidak boleh hanya bergantung pada hasil hutan sebagai satu-satunya sumber pendapatan mereka. Kemahiran komunikasi mereka perlu dipertingkatkan supaya komuniti orang asli di Tasik Chini dapat melibatkan diri dalam aktiviti pelancongan seperti menjadi pemandu pelancong di Tasik Chini atau menubuhkan rumah penginapan seperti yang telah dijalankan oleh Rajan Jones Guest House atau modal sendiri dan dibantu oleh kerajaan. Aktiviti eko pelancongan yang berasaskan kebudayaan orang asli ini perlu dipertingkatkan dan diperluaskan bagi meningkatkan kembali minat pelancong ke kawasan ini. Seperti semua sedia maklum, Tasik Chini merupakan tasik semula jadi yang kedua terbesar di Malaysia. Tasik Chini ini yang terletak di Mukim Penyu merupakan sebuah kawasan yang tempatan banjir semula jadi yang penting bagi lembangan Sungai Pahang. Angin monsun timur laut yang telah melanda baru-baru ini telah mengakibatkan hujan lebat dan kenaikan paras air di negeri Pahang dan Johor, terutama di kawasan Chini yang juga tidak terkecuali. Cuaca buruk ini telah mengakibatkan banjir di kawasan rendah dan secara tidak langsung menenggelamkan rumah-rumah penduduk yang berdekatan dengan paya, tasik dan sungai. Penempatan orang asli berdekatan dengan Tasik Chini telah dinaiki air seperti Kampung Tanjung Puput, Kampung Melai dan Kampung Cendahan. Tidak dinafikan monsun ini merupakan kitaran hidrologi semula jadi, namun sudah tentu aktiviti guna tanah yang tidak terkawal menentukan skala impak negatif yang dibawa monsun ini. Oleh itu saya sangat menyokong usaha pemeliharaan dan pemuliharaan yang dijalankan di Tasik Chini bagi mencapai idea-idea berkenaan dengan pengurusan secara lestari dan berkesan. Penempatan orang asli yang berdekatan dengan Tasik Chini, khususnya di Kampung Puput dan Kampung Cendahan ini, kita perlu dikomersialkan sebagai perkampungan budaya orang asli yang akan menjadi pilihan destinasi pelancong ke Tasik Chini. Kedua-dua kampung ini lagi mengekalkan konsep kampung budaya dan mempamerkan hasil kraf tangan orang asli untuk dijual kepada pelancong yang datang berkunjung. Hasil kraf tangan ini harus diperkembangkan di atas talian bagi mempromosikan ekonomi hasil tangan orang asli di Tasik Chini ini. Pada pihak saya amat berbesar hati untuk terlibat dengan aktiviti seperti ini. Saya amat berharap pihak PPTC dan juga penganjur dapat menjadikan program ini sebagai aktiviti tahunan. Sekiranya terdapat mana-mana pihak yang memerlukan bantuan dan sokongan saya amat berbesar hati untuk menghulurkan bantuan semampu mungkin. Saya juga amat besar hati sekiranya pakar-pakar penyelidik dan ahli akademik dapat mengusulkan kepada saya cadangan-cadangan untuk saya kemukakan kepada pihak kerajaan dalam usaha membangunkan DUN Tasik Chini. Cadangan daripada semua pihak amat saya alu-alukan. Akhir sekali, saya amat berharap persidangan yang dihadiri oleh ahli akademik global, pemain industri dan golongan muda ini dapat menyebar luaskan lagi kepentingan alam sekitar. Selain itu, besarlah harapan saya bahawa persidangan ini dapat menjadi pemangkin kepada ahli akademik dan pemain industri dalam menggalakkan pembangunan ekonomi masyarakat setempat dan tidak mengabaikan keadilan kepada alam sekitar. Akhirnya saya sudahi dengan wabillahi taufiq wal hidayah. Assalamualaikum warahmatullahi wabarakatuh.
Thank you, YB Muhammad Syarim. We hope all the activities and programs arranged by the local authorities will help in promoting Tasik Chini as tourist centre and at the same time be able to raise the local community socio-economy, such as promoting their cultural village and unique handicraft. Ladies and gentlemen, are you ready for our next video? Let's hear from Miss Deshin Anak Perempuan Kota Dapani from Universiti Kebangsaan Malaysia.
Hello everyone, my name is Dashini, Daughter of Good Dapani, and my topic for today is self-awareness regarding Tasik Chini as Biosphere Reserve and its importance. I believe that even today, there are still many Malaysians who are not mindful of what's happening to Tasik Chini. In fact, I was left shocked when I read some news online regarding Tasik Chini that was once a beautiful and unique wildlife reserve, now is leading to its destruction. So, let's get to my point.
Firstly, the major problem that promotes destruction to the freshwater lake is the human activities in its surrounding. Logging and mining without proper land rehabilitation leads to soil erosion, which pollutes the water after every rainfall. This causes the once clear water now turns cloudy. Besides, Tasik Chini is known for its rich diversity of flora and fauna and is home to various species of fishes, birds, and aquatic plants. These species of flora and fauna are something that we can hardly find or cannot be found at all elsewhere. This is what makes Tasik Chini unique on its own. Thus, if humans persist with those destructive human activities, 10 years later, all of these species will be threatened by extinction. It will be a pity for the future generation since they will not experience and enjoy the uniqueness of the natural flora and fauna habitat found in Tasik Chini. Humans should be aware of the long-term effects of such action on the well-being of Tasik Chini. Despite of UNESCO's Biosphere Reserve status, it doesn't seem to stop humanity from performing ecologically destructive activities. Tasik Chini should be protected to be brought to the future generations as it is. Furthermore, there are about 800 indigenous people living in villages surrounding the lake, and they depend on the lake for livelihood and water supply. Where else would they get clean water when the lake becomes polluted? Even fishes are not edible since they are contaminated and can be hazardous to the orang asli's health.
In conclusion, Tasik Chini needs to be saved today from further destruction, and effective measures need to be implemented to re-establish Tasik Chini to its primordial condition. I hope that the government takes this issue in hand, since they are the highest authority which can resolve this problem. That's all from me. Thank you.
Thank you very much, Ms. Dashni. There's a lot of local people depends on the lake for their livelihood and water supply. Therefore, it is important to keep the lake from the further destructions. Alright, ladies and gentlemen, it is my pleasure to welcome our next presenter. Please welcome Dr. Ranjita Pubati from Quest International University, Malaysia, to present on her research, induction of photocom-like bodies from thin cell layers of Ludisia discolor. Please welcome Dr. Ranjita.
Hello everyone, I am Dr. Ranjita Pubati from Quest International University Perak, which is located in Ipoh. And today I will be presenting a piece of my research entitled The Induction of Protocom-like Bodies from Thin Cell Layers of Ludisia discolor. Ludisia discolor is an orchid plant. Orchids are typically propagated from seeds. Each seed can be found in the capsule, which contains millions and millions of seeds. So each of the seed will ultimately form protocoms. Protocom-like bodies do not form from seeds. They can be found on either tissues or explants of the orchid plant. Initiation is pretty simple. They originate from callus cells, which form compact regions composed of either meristemoids or promeristems, which are highly regenerative tissues. Protocom-like bodies also display polarized growth, and they emerge from the surface cells of each compact pool of meristemoids. Jewel orchids belong to the tribe Cranicidae and subtribe Goodyearinae. They typically thrive under very warm conditions. These orchids are prized for the leaves rather than the flowers. The flowers are mostly white in color, but the leaves are highly colorful, usually displaying pink, red, or purple hues, and are laced with silvery veins or even golden veins. Ludisia discolor used to be known as Hemaria discolor. It typically grows in damp places under tropical temperatures. It thrives under very low light intensities because direct sunlight actually decolorizes the leaves. Basically, the technique capitalizes on the excision of minute-sized explants from various plant tissues. Plant tissues can be either the leaves, stem, or even roots. There are two types of thin cell layer techniques here. You have the longitudinal TCL, which is known as the LTCL, and then you also have the transverse TCL, which is the PTCL. Orchids are typically propagated using their stem segments. The stem region is usually sliced into half and then placed directly on the medium. The TCL root employs the very minute thin slicing of the stem, leaf, or root regions, after which all of the segments will be planted on the medium. So the TTCL root employs the cross sections of the stem or the tissue, after which all of them are planted on the medium. And the LTCL root employs longitudinal sections of the tissue, after which again all of the segments will be placed on the medium. So the numbers of new tissues that are formed depend on the surface area of the tissue which is exposed to the medium. That is the crux of the TCL method here. With that, the objectives of this research was to induce PLBs from transverse TCLs of leaves and nodal segments of Ludisia discolor. This is followed by the observation of the developmental stages of PLBs, which are generated from TCLs of Ludicea discolor through freehand sectioning, toluidine blue O staining, and light microscopy. In order to perform this research, the plants were sourced from the School of Biological Sciences, University Science Malaysia, and were maintained in half-strength semi-solid MS medium, supplemented with all of these components here. The plants were cultured under 16 hours photoperiod under cool white fluorescent lamps with the temperature set at 25 plus or minus 2 degrees Celsius. For this research, the parameters tested included the type of medium used, which are half-strength MS medium, half-strength Mitra medium, as well as the use of plant growth regulators such as NAA, BA, 2,4-D, and PDZ. In terms of the composition of medium, the MS components present a slightly different composition when compared to Mitra components, in that the amount, the concentration of ammonium and nitrates which are present in the medium is reduced drastically in the Mitra medium when compared to the MS medium. The Mitra medium also contains a few vitamins which are not found in the MS medium. For the stem segments, a total of 7 treatments were employed involving various concentrations of plant growth regulators. While in the case of leaves, a total of 5 treatments were employed involving the use of all of these plant growth regulators as well as these concentrations of PGRs. In order to prepare thin cell layer, all you need is a razor blade. Basically, all you need to do is to remove the plant from the culture, cut the plants into super small slices, after which you use a razor blade to further slice the explants into transverse thin cell layers. You can use a graph paper to aid you in this endeavor. In order to observe the growth and development of the PLBs from the TCLs, we employed the toluidine blue O staining method. This was to observe the PLB structure, the vascular section of the PLBs, as well as the connection between the PLB and the stem region of the transverse thin cell layers. This is very, very important in order for us to make the distinction between PLB formation and shoot formation in all of the explants that we tested. So the images were captured using a light microscope affixed to a color video camera and then analyzed using CellSense imaging software. In terms of data analysis, weekly observations were made for the following parameters, which are the number of weeks for PLB induction, number of weeks for plantlet formation, as well as root hair formation in all of the explants. In terms of statistical analysis, all data were analyzed using either one-way, two-way, or three-way ANOVA, with the means differentiated using 2K's honor significant difference test. All data analysis were performed using SPSS version 22. For the results, it was observed that medium did indeed influence the production of PLBs from TTCLs of Ludicea discolor. It was observed that 1 milligram per liter BA produced the highest number of PLBs, and this was followed by 0.50 milligram per liter of BA, as well as 0.75 milligram per liter of NAA. In terms of plantlet production, no significant differences were found between the means obtained for all the medium and plant growth regulators tested in this study. In the case of root induction, observations obtained were similar to that for plantlet induction for all stem TTCLs tested in this study. Data from the use of leaves were eliminated from the study as all the treatments did not produce any results. In the case of three-way ANOVA for all the parameters tested, it was found that there was an interaction between the media, plant growth regulator, and PGR concentration in producing PLBs from the TTCL that were cultured in Mitra medium. PLB development can be obtained as a small bump that originates from the 3rd week of study on all stem TCLs that were cultured in either Mitra medium or even MS medium. The bump will form protocom-like bodies that have a bipolar nature to their structure, and this will ultimately differentiate into shoot and roots, and finally an orchid plantlet. Figure B shows the shoot development as well as root development that can be found on the transverse tinsel layer of the Rhodesia discala stem that were cultured on the Mitra as well as MS medium. Rhizoids naturally developed on all segments regardless of the treatments that were employed. PLBs that were obtained from the TTCL experiments were subjected to toluidine blue staining and further observed under the light microscope. It was observed that the globular-shaped PLBs, labeled as P over here, formed directly from the stem TTCLs, labeled as S, which is complete with their own vascular system and shoot apical meristem here. The vascular system of the induced PLB was also found to be directly connected to that of the stem TTCLs. Ultimately, as the PLB progresses in size, it will detach itself from the mother plant, which in this case is the stem TTCL of the explants that were all placed in the medium. The TTCL technique has been successfully used to induce PLBs in a number of orchid species, such as Dendrobium nobile, Cymbidium aloifolium, and also Cymbidium sweeping neem. It is found that the addition of cytokinin is an important step in enhancing PLB induction. The biggest asset of the TCL technique is generally its ability to increase the surface area upon which PLBs can be formed. Apart from that, it was also found that ammonium sulfate and potassium nitrate, when present in higher amounts, are actually inhibitory for protocol formation. In the case of Mitra medium, the components were present in a lesser amount, which actually facilitates the growth and development of protocol-like bodies. It was also found that PLBs can be induced in two ways, via direct embryogenesis from either stem, leaves, shoot, and root tips, which is actually observed in this study. PLBs can also be formed through indirect embryogenesis, in which a callus intermediate stage is involved. In this study, it was found that PLBs were induced from the stem TTCLs without any intermediate callus stage. It is very important to avoid the callus stage in propagating certain breeds of orchids because we do not want variability in terms of the genotypic and phenotypic expression, which can occur in callus cultures. The leaves of Rhodesia discala failed to induce the PLBs in both the medium. Generally, it was observed that mature leaves will produce poor response when it comes to orchid propagation. To conclude this study, it was found that the best treatments for inducing PLBs from stem TTCLs of Rhodesia discala is generally Mitra basal medium combined with either 1.0 mg per liter BA, 0.5 mg per liter BA, or 0.75 mg per liter NAA. The PLBs that were formed in this study were found to be globular shaped, was induced within 6 weeks of treatment, and were induced directly from the stem TCLs without an intermediate callus formation. I would like to thank Quest International University for supporting this study. I would also like to thank Prof. Dr. S. Sri Ramanan from University Science Malaysia for providing the plans as well as support for this study. And last but not least, I would also like to thank Temuli Shanmugam for carrying out this study on my behalf. Thank you all for your time, and I wish all of you a good day ahead. These are some of the references that I have employed for this study. Thank you very much again.
Thank you very much, Dr. Ranjita Bhawati, for sharing us your interesting technique. It is my privilege to introduce our next keynote speaker. Please welcome Tuan Baitzawi Chemat, Chief Executive Officer of the East Coast Economic Region Development Council ECRDC, to give a talk about comprehensive multi-skill approach in achieving sustainability in Tasik Chini. Please welcome.
Assalamualaikum and a very good afternoon. Before I begin, let me introduce myself. My name is Baitzawi Chemat. I'm the CEO of the East Coast Economic Region Development Council, ECRDC. I'm glad that despite the unique circumstance that we are all in today, We are able to virtually participate in the e-conference as champions of Tasik Chini and locals in the area. I will start by explaining about ECRDC before speaking on my topic for today, which is comprehensive multi-scale approach in achieving sustainability in Tasik Chini. The East Coast Economic Region, also known as ECER, covers the states of Kelantan, Terengganu, Pahang, and the district of Mersing in Johor. It is home to some 4.8 million people, or about 15% of the total national population. ECRDC is a statutory body established to spearhead the execution and implementation of the East Coast Economic Region Master Plan, which plays a lead role in setting the direction, policies, and strategies for the socio-economic development in ECR by promoting and facilitating investment into the region. The vision of ECR is to be developed region by 2025. We are now implementing our second master plan, the ECR Master Plan 2.0, from 2018 to 2025. The ultimate objective of ECR's establishment is to ensure a balanced regional development in the ECR, thereby uplifting the quality of life of the people in the region, taking into consideration three important indicators. The first one is people economy, the second one is capital economy, and the third one, natural endowments. One of the catalysts for economy is tourism, and the ECR is blessed with diverse tourism attraction from island to ecotourism location. It is also the melting pot of Malaysia's heritage and culture, which include traditional performing arts and handicraft. This is why ECRDC applies the rainforest to reef concept, R2R, to create a different experience for tourists by bringing together the natural assets of a location. Leveraging on these natural and cultural assets, as well as other untapped opportunities, ECR is set to accelerate the region economic growth in a viable, equitable, and sustainable manner in alignment with the United Nations Sustainable Development Goal, SDG. Therefore, tourism development in the ECR is undertaken in a manner that ensures the sustainability of the natural environment. One of the key attractions within ECR is Tasik Chini in Pekan Pahang. In May 2009, Tasik Chini made history by becoming the first ecological site in Malaysia to be recognized as a UNESCO Biosphere Reserve, one of only 669 locations around the world to receive such a recognition. The recognition was bestowed only to areas comprising terrestrial, marine, and coastal ecosystem that showcases the signs of sustainability. Recognizing the uniqueness of Tasik Chini and its strong ecotourism potential, ECRDC in 2009 had proposed Tasik Chini to be gazetted as a state park. The status and recognition are highly important as they pave the way for more research-driven conservation and preservation efforts. Not only does this help to protect the beauty of Tasik Chini, it will also act as a reference for other ecological sites in Malaysia. ECRDC, in partnership with Pahang State Government, the Ministry of Energy and Natural Resources, KETSA, and University Kebangsaan Malaysia, UKM, have been working together for almost 10 years to support the government's commitment to conserve and preserve Tasik Chini, Malaysia's first UNESCO Biosphere Reserve. ECRDC vision for Tasik Chini is for it to become a world-class ecotourism destination which protects the biodiversity, culture, community, and other assets via sustainable tourism development principles. There are three main pillars that need to be kept intact, which are conservation and preservation, community development, and sustainable tourism, with physical infrastructure and programs to align with Tasik Chini with Man and Biosphere Reserve, MAB status. In order to achieve these visions, 3 key actions have been determined. First one, to implement an immediate action plan as well as efforts to conserve and preserve Tasik Chini. The second one, to further strengthen the role and image of Tasik Chini as Malaysia's first biosphere reserve. And last but not least is to increase education, awareness and public involvement in these conservation and preservation efforts. In 2009, IECSBR embarked on the Strategic Implementation Plan, SIP, for Tasik Chini to reaffirm our commitment to conserve and preserve Tasik Chini. The SIP covers key initiatives and strategies up to 2025 via a 3-phase approach. There are 71 projects and programs that have been identified in the SIP. One of the key measures under the SIP is the establishment of 3 management zones of Tasik Chini according to the Biosphere Reserve framework. They are the core, buffer and transition zones, each with varying levels of protection and human activities. Now let me share with all of you our initiative in Tasik Chini since 2009. IECSBR spearheaded the implementation of conservation, preservation and integrated management plans, as well as the gazetting of Tasik Chini State Parks, along with Tasik Chini Research Centre, or PPTC UKM. IECSBR and UKM are strategic partners that help each other to ensure the sustainability of Tasik Chini. In 2010 until 2012, the immediate action plans for conservation and preservation have been implemented with 4 key initiatives which I will explain later. In 2011, we started implementing the Strategic Implementation Plan, or SIP, for Tasik Chini. Physical construction began in 2012 with the construction of freshwater laboratory complex consisting of 3 main laboratory buildings for in situ monitoring and research for academicians, while deepening understanding of Tasik Chini among the locals and tourists. The next phase of construction focused on the supporting building to the main labs such as accommodation, community square and jetty. I would like to highlight that all development was carried out holistically and continuously to ensure Tasik Chini sustainability. One of our most important efforts in installing water quality monitoring station in major feeder rivers to Tasik Chini as a continuous effort in water quality monitoring, as well as to enable hydrological and meteorological data logging. The presence of the monitoring station has kept the water quality of Tasik Chini in check at all times. The station are able to look at the hydrological system and water levels in real time. The construction of the station is important to ensure that we can achieve Class 1 in water quality. This will be a landmark achievement as the water in Tasik Chini will be safe for consumption without any additional treatment except for boiling. In Tasik Chini, fish community also plays an important role in balancing ecosystem in the lake. Following our initiative in reintroducing selected native fish species like lampam, sebarau, and kelah through an aquaculture approach, the fish population in Tasik Chini is increasing and thriving. At the same time, IECSBR also carried out the installation of various river bank revetment along Sungai Chini such as fiber roll geotextile, gabion, and artificial levee to help reduce riverbank erosion problem. Coconut coir logs are also used in Sungai Chini. Coir logs are erosion control product which is made of 100% compressed biodegradable coconut fibers. Meanwhile, Sungai Jemerau and Sungai Melai are now free from the breeding of cattails or pokok ekor kucing for locals and rasau trees which were overpopulated within the lake's water body. These 2 aquatic plants caused nuisance to boat navigation and competed with native species. These 2 species are also potentially invasive and threaten the ecology and open slow-flowing water bodies. They also competed with second lotus that is unique to Tasik Chini. These continuous efforts and monitoring via the 7 water monitoring station, river cleaning, and riverbank erosion initiative have rejuvenated Tasik Chini and resulted in the improvement of the water quality to Class 2 from the previous Class 3 in 2008. Apart from our rehabilitation and conservation efforts, IECSBR also carried out physical development. The main construction in Tasik Chini is the freshwater laboratory complex. This research complex, which was developed using the green building concept, was constructed on a 10-acre area in the southeast part of the Tasik Chini Biosphere Reserve, TCBR. The first phase consists of 3 laboratory buildings, namely hydrology and climatology, instrumentation and microbiology, and water analysis laboratory, as well as administrative office, boardwalk, and guardhouse. The research complex was handed over to UKM on January 2014 and is being maintained by the university. The second phase of construction comprised of hostel for researchers and other ecotourism facilities, including the Orang Asli Square, which were completed in 2017. It now serves as a hub for all research, education, and monitoring activities within the TCBR, in line with the Tasik Chini vision and goals to rehabilitate and conserve the lake as well as its ecosystem and biodiversity. The interesting part of the laboratory complex construction was that it was built based on low ecological impact design, which is an environmental friendly method. The physical structure also blends well and coexists with the native trees. The boardwalk was also built on stilts to reduce impact to the ground. While conservation and preservation initiatives are still ongoing, much progress has been achieved in Tasik Chini. For Phase 3, we have embarked on the Lotus Conservation Project together with Forest Research Institute of Malaysia, or FRIM. Lotus is an icon of Tasik Chini. Therefore, the preparation of planting stock is an important element to ensure that lotus can be planted at any time, even after the flood season. However, this is not an easy task, as preparation of planting stock needs to follow the appropriate methodology to ensure its sustainability. The project is still ongoing until year 2023. As a conclusion, Tasik Chini is important not only to locals and to the people of Pahang, but to all Malaysians. It is both a tremendous gift and great responsibility. Together with our many partners, we are working to protect and restore the sustainability of this legendary lake. We hope to return Tasik Chini to its pristine state, a self-sustaining bioeconomic ecosystem with a positive contribution to nature, livelihood, culture, and social economy to the local community. Ultimately, we must realize that all of us have a role to play in conserving and preserving this natural heritage. Only by working together can we ensure that our children will inherit the legacy of a healthy and resilient ecosystem for Tasik Chini. As a final say, we have made significant improvements and we are on track, but there is still more to be done. Let us all work together to retain Tasik Chini's UNESCO Biosphere Reserve status. With this, I end my talk. Thank you very much.
I would like to give my hearty vote of thanks to our Honourable Keynote Speaker, Tuan Bakzawi Cik Mat, for gracing to this conference. Thank you, sir, for your very thought-provoking and interesting address.
Yes, alright. If you have any question to ask during any session, type your questions in the comment below box, the session video, and click post comment button. Alright, and we will to get question to be answered by our community. Alright, ladies and gentlemen, see you later.